Hi, my name is Doug Huang, and today I'm going to be presenting on muscular growth and genetics for Dr. Denison's genetics class. Muscles are divided into cardiac, smooth, and skeletal muscle. I am focusing on skeletal muscle. There are 650 muscles in the human body, so it's important to understand how they function and grow. Muscles provide protection, movement, and temperature regulation. Muscles are composed of cells called myocytes. Myocytes are composed of bundled fibers called myofibrils, and myofibrils are composed of myofilaments. A functional unit of myofibrils within a muscle is called a sarcomere. As you can see in the diagram, I'm going to be focusing on the skeletal muscles like the pectorals, the biceps, the rectus abdominis, which are the abs, and the other things that we can train to grow bigger and stronger. Nerves control muscular function at the neuromuscular junction. An action potential triggers the release of acetylcholine, a messenger molecule. Acetylcholine binds to receptors on the cell's sarcoplasmic reticulum, triggering the release of calcium ions from within its membranes. Release of calcium ions allows for muscle contraction. Myocytes have bundles of myofibrils composed of thick myosin filaments and thin actin filaments. Calcium ions attach to tropin molecules and cause tropomyosin molecules to reshape, exposing actin binding sites. ATP energy transfer causes myosin to bend and attach to the actin molecule, forming a cross ridge by which actin and myosin are pulled closer together. Myocytes have bundles of myofibrils that are composed of thick myosin filaments and thin actin filaments. Calcium ions attach to tropin molecules and cause tropomyosin molecules to reshape, exposing actin binding sites. ATP transfer causes myosin to bend and attach to the actin molecule, forming a cross bridge by which actin and myosin are pulled closer together. This combined muscle contraction allows for the flexion of muscles. Hi, my name is Doug Huang, and today I'm going to be presenting on muscular growth and genetics for Dr. Denison's genetics class. Here is a muscle diagram. Muscles are divided into cardiac, smooth, and skeletal muscle. I am focusing on skeletal muscle. There are 650 muscles in the human body, so it's important to understand how they function and grow. Muscles provide protection, movement, and temperature regulation. Muscles are composed of cells called myocytes. Myocytes are composed of bundled fibers called myofibrils, and myofibrils are composed of myofilaments. The functional unit of myofibrils within a muscle cell is called a sarcomere. As you can see in this diagram, I'm going to be focusing on muscles like the pectorals, biceps, abs, obliques, quads, glutes, and other muscles you can grow through muscle hypertrophy and exercise. The neuromuscular junction. This is how muscles work at the molecular level. Say I want to move, my brain triggers nerves, causing a chain reaction of action potentials that cause muscle flexion. Nerves control muscular function at the neuromuscular junction. An action potential triggers the release of acetylcholine, a messenger molecule. Acetylcholine binds to receptors on the muscle cell's sarcoplasmic reticulum, triggering the release of calcium from within its membranes. Release of calcium allows for muscle contraction. Muscle function. Myocytes have bundles of myofibrils composed of thick myosin filaments and thin actin filaments. These actin-myosin complexes pull their movement together, called the sliding filament theory. When we flex a muscle, calcium ions attach to tropin molecules and cause tropomyosin molecules to reshape, exposing actin binding sites. ATP energy transfer causes myosin heads to bend and attach to the actin molecule, forming a cross bridge by which actin and myosin are pulled closer together. This combined sarcomere contraction with millions of cells allow for muscle flexion. This is how muscles grow. Muscle cells are provided by veins and capillaries that carry nutrients. After muscle damage through training or exercise, cytokines and chemokines trigger the chemotaxis of satellite repair cells. These satellite repair cells fuse with the muscle cell, producing a myonucleated muscle cell. This causes the expansion of muscle cells and increased actin-myosin development. Over time, the cells will increase in size, and the flexion of the muscles will produce greater forces. Here is a molecule that is integral to the growth and differentiation of muscle cells. It's called MyoD, or Myoblast Determination Protein 1. MyoD differentiates stem cells and satellite cells into myoblast cells, which are cells that produce muscle cells. MyoD is also involved in muscle repair. MyoD is a transcription protein that binds to the E box. The E box is an enhancer box that binds transcription factors and facilitates transcription. Its consensus sequence is the palindromic CACGTG. MyoD is located on chromosome 11, band 11, arm P15.1. MyoD production is enhanced after exercise and muscle damage. High levels of MyoD causes cell renewal and differentiation. However, damage to the MyoD gene may not reduce muscle development drastically because of functional redundancies with MyF and MRF4, which are similar transcription factors that activate muscle genes. PAX3 activates MyoD transcription. MyoD binds to the enhancer of the myogenin gene. The production of myogenin is activated. Myogenin goes on to activate the production of other proteins. The proteins activated by myogenin are actin, myosin, troponin, and tropomyosin. So once this gene is triggered, it causes the production of proteins that further the differentiation and just causes a domino effect that produces more and more muscle proteins. Moreover, MyoD is more involved in the development of fast-twitch muscles. MyoD transcription function. So the function of MyoD is regulated by something called CAP1. CAP1 is a protein involved in transcriptional regulation, cellular differentiation, and proliferation, DNA damage repair, viral suppression, and apoptosis. CAP1 binds to coactivators P300 and LSD1 and co-repressors G9A and HDAC1, forming a complex at the promoting regions of muscle genes that prevents transcription. MSK1 phosphorylates CAP1 and allows for MyoD activation. MyoD triggers chromatin remodeling at the E box. MyoD binds at hundreds of muscle gene promoters. Interferon-related development regulator 1, IFRD1, is a positive cofactor of MyoD. IFRD1 and MyoD co-activate MEF2C, myocyte-specific enhancer factor 2, by displacing histone deacetylase 4. NAFTC1 is a transcription factor that inhibits the physical interaction between P300 and MyoD, preventing transcription. This inhibition allows for the differentiation of slow-twitch muscle fibers.